Hello students, today we will study about the secondary growth in plants and we will study secondary growth in stem. Firstly, primary growth of plant is responsible for growth and development of plant. प्राइमरी ग्रोथ से प्लांट केवल हाइट में बढ़ता है जबकि सेकेंडरी ग्रोथ से प्लांट जो होता है उसका डायमीटर बढ़ता है. इट मीन्स द स्टेम इंक्रीजेस इट्स विड्थ ड्यू टू द सेकेंडरी ग्रोथ. And the primary growth of the plant takes place with the help of shoot and root apical meristem. It means the primary growth of the plant takes place like this. That shoot has apical meristem at its apex, and the roots also has a apex, apical root apical meristem. So that's why the primary growth of the plant takes place with the help of shoot and root apical meristem. This apical meristem consists of meristematic cells, and due to these meristematic cells, it regularly increases, divide, and afterwards, the shoot of the plant increases in height. In this way, root of the plant increases with the help of root apical meristem. Whereas if we see in secondary growth, it takes place with the help of the cambium. Cambium is a meristematic, secondary meristematic tissue, and it has also the power of division, so it regularly divides when secondary growth takes place in plant. Herbaceous monocots and herbaceous eudicots do not show any secondary growth because these do not consist of these vascular cambium. Gymnosperms and woody dicots increases in width due to the secondary growth because there is uh, vascular cambium in the stems of these dicots and also the gymnosperms. Now come to the next, that the cambium is of two types. Normally the cambium is of two types, vascular cambium and cork cambium. Vascular cambium is found in vascular bundles. It means we can see the structure of stem. We can see the TS of the stem, and it shows that there are bundles, vascular bundles, which are arranged in a ring, and there is xylem and there is phloem, and between this there is a cambium cells. These cambium cells have the power to divide again and again when secondary growth takes place. The vascular bundles of dicotyledon system arranged in a ring, they are collateral and open divascular bundle. They contain a single layer of cambium cell which separate the xylem and phloem. This point to be understood very clearly, that the xylem and phloem, the cambium present between the xylem and phloem, it is known as the fascicular cambium. And on the other hand, the the cambium which is found between the two bundles, that is known as the interfascicular cambium. So there are two type of cambiums, interfascicular and fascicular cambium. Fascicular cambium is that cambium which is found between the xylem and phloem in a bundle, and interfascicular cambium is that cambium which is found between the two bundles. And with the help of this interfascicular cambium and this cambium, a ring-like structure formed in the stem during secondary growth. Now this is, you can see here the cambium cell. This is one cambium cell, and it will divide during secondary growth into a phloem cell and xylem cell. In this way, it will divide regularly, and one phloem cell and two xylem cell. Two phloem cell, and we will see three xylem cell. It means the division takes place regularly, and xylem cells are in more number than in phloem cells. It means the tissue of xylem becomes more in comparison to phloem. Now you can see the structure of TS section of stem that consists of a vascular bundle, and the secondary growth is taking place, and secondary growth takes place in these bundles like this. A ring like structure of cambium has formed here, and in this way the you can see the width of a stem, it is increasing because xylem, a secondary xylem is here, and secondary phloem is developing, and secondary xylem and secondary phloem is developing regularly, and this is a cambium ring. This cambium ring divides regularly and form a xylem, secondary xylem inside and secondary phloem outside. In this way the stem become so thickened. So, and you can also see the medullary rays are there. How the medullary rays are formed? The cambium in starting consists of a fusiform initials, and the other one is ray initial. Fusiform and ray, and the other one is ray initial. Ray, R-A-Y, ray initials. With the help of these ray initials are cells which are oval in shape, and these are also parenchymatous. So it divides regularly in this area. Ray initials divide regularly and form a ray-like structure of parenchyma, and it opens outside. From here, it starts and it open outside, and these are ray initial cells which perform this activity. In this way these medullary rays are formed, and we can call it medullary rays. In every diagram you can see in the stems we can see medullary rays. So in this way the secondary growth takes place in the stem. Now what happens about the phloem? What is phloem? For first point is this: when a stem is increasing in size and there is a secondary growth taking place, so the xylem tissue and the phloem tissue is increasing regularly. So what happens with the increasing of xylem and phloem tissues, the outer portions become so compressed with the tissues which are found in between, which are found in the area. Xylem tissues and phloem tissues are so much formed in the stems during secondary growth that they compress the outer tissue, and there we can see the cork cambium. This cork cambium outerward side, this cork cambium is found outerwards, and we can see here that these are the cortex, and here we can see epidermal layer, and with the help of this epidermis some cells become cork cambium, and these have the power to divide. These divide into two: one is a cork and other one is phalloderm. Can inner side phalloderm and outer side it forms phallum. So we can understand it clearly that cork cambium is also known as a phloem, and this phloem will divide into two: one is a phallum outer side and other one is phalloderm. And what is phloem? Phloem is a cork cambium. It has a power to divide, and it consists of cork consists of suberin lignin, and in this way the bark of the plant forms, and it regularly formed, and it also crushed outside, and inner side phalloderm will form the secondary cortex. And we know that the cells of the cortex regions are parenchymatous. So phloem, phallum, phalloderm, three of these are known as periderm. These three are known as cork cambium, and there is phalloderm also. These three are called as periderm.